Thanks, Tim. So this is actually my first ability to speak with outside of Teams and Zoom, so it's a little weird, but really excited about it. In fact, we just had one of our big meetings for what I study, the Shock Society meeting. We were supposed to be in Portland, and that was recently canceled. So now I'm sad to actually have to do a Zoom meeting. But as Tim mentioned, I've known him quite a while. We began using Luminex for cytokine assays. Have a Luminex. We actually have a Luminex 200, which is now in storage. MagPix, and then the XMAP as well, which we used for all of the COVID research that I'm going to be presenting. And now it looks like we're going to have to raise some money to get a new system because it looks better. Okay, so as Tim mentioned, our labs at Harvard basically shut down on March 13th. At the time, I was, we were just focused on getting everyone kind of back remotely, and started working on research projects and writing. But then what occurred was all the news came out about all these really terrible antibody assays, and that it seemed as though they needed some help. So I was dragged into, and very happily dragged into, the Massachusetts Consortium on Pathogen Readiness. This was formed early on in the pandemic, and this interaction really led to me returning to the laboratory. I was the only one that was allowed to actually go back to my lab, so I was actually working solo in the lab. So, as a PI, sometimes it's kind of fun to just get your hands back at the bench. So all this work really started right at the beginning, within a month after the pandemic started, and it all came about because of the Massachusetts Consortium on Pathogen Readiness, and also interactions among all the scientists in Massachusetts and Harvard that really wanted to help and get involved in understanding the infection, helping with vaccinations, treatments, and all the important things that have led to improvements today. So these are the two guys I really started working with during the pandemic. The first person is Dwayne Wesseman, who's a B-cell specialist, antibody specialist, who worked with Fred Alt. Early on during these meetings, we were talking about developing assays. He was doing a lot of ELISA assays. I had in my laboratory a huge collection of plasma samples that were pre-COVID based on our immunology research. So I began sharing samples with Dwayne. And because of this, then I met Steve Elledge, because Dwayne said that he was working with this guy who was developing a platform that would identify all the viral epitopes that are recognized by antibodies in plasma. So what occurred from this, through weekly Zoom meetings, which were really designed to share and discuss technologies, it led to a number of collaborations, not only collaborations with Dwayne and Steve Elledge, but collaborations with Denisa Wagner, who's interested in neutrophils. The paper is hopefully going to get published very shortly, where we're looking at net formation in the lungs of patients that were suffering from COVID disease. So as I mentioned, Dwayne was really, he's a specialist at antibodies. I learned a ton from him about the virus and the antibody responses. And then Steve, of course, is an expert in developing platforms for screening for antibodies. And in particular, he's very famous for his technology called VirScan, which I'm sure some of you have heard about, but it's an approach for basically taking any virus sequence and putting it into a bacteriophage, which would then allows you to probe a very high throughput for antibodies that are binding to the specific peptides of a virus or different viruses. So as Tim mentioned, all this work was a large collaboration with many people in Massachusetts, but in particular, I worked very closely with Steve Elledge's lab to develop these Luminex assays that we eventually used to look at to look at the SARS CoV-2 specific reactive antibodies against peptides. So if you want to take a closer look at this work that I'm going to be presenting, here's the reference. Okay, so early on in the pandemic, there was a lot of exchange of information, and in particular, the sequence of SARS CoV-2 was released as soon as it was identified in China. From this, there was a flurry of activity where everyone began making proteins and different reagents for assays. And Steve was one of these people. He was very much working with the virologists to develop these libraries, which he used in his VirScan technology. So I'm going to do my best to describe his technology. I did not perform these assays, but it's really kind of a neat approach that they do. So what they first do is they take the viral genome and they basically develop a DNA library of all the open reading frames with different overlapping sequences, which would be either 20 mers with a 5 amino acid overlap or 56 mers with a 28 amino acid overlap. These are then, as I mentioned, synthesized into a library, which we then put into the T7 bacteriophage, and then they display these on E. coli. From this, what they do is then they add serum antibodies or plasma antibodies, and they incubate these antibodies with the E. coli, which express the specific peptides. And then after that, they perform an immunoprecipitation reaction, which allows you to basically concentrate those E. coli that are expressing peptides that are specifically reactive with the antibodies. Now, you'll note that his assay really focused on IgG and IgA. Of course, there's other types of antibodies made like IgM, but it's well known for those of us that are working with these immunoassay platforms and with immunoprecipitation that IgM is very sticky and provides a lot of background. And of course, IgM is your primary antibody, so looking at IgG and IgA is really kind of the focus of this technology. So then what he's done is they have a barcoding approach with PCR, which allows them to deconvolute which of the actual clones of the T7 clones is within each E. coli. They perform sequencing, and then they can figure out which peptide is being recognized by the antibodies. So this is the basis of this VirScan technology. He's obviously, this has been his bread and butter approach, not only for antibody detection for peptides, but he's also used it for T cells and also class 1 restricted T cell antigens and also class 2 restricted responses. Go for it. Okay, so starting out, this is a phylogeny tree of 50 coronaviruses that have been identified. And I don't know if it shows up very well here, but those that are identified in red are the actual viruses that he included in his VirScan platform. And shown kind of at about, I don't know, 10 o'clock or so, 9, 10 o'clock, you can see that there's the beta coronavirus B, which includes many of the coronaviruses that were of concern and are of concern during the pandemic. And then on the upper quadrants there are the alpha coronaviruses that are included, and these are the common cold viruses that we've all been exposed to. which are not, they're pathogenic, but they're not as deadly as the SARS CoV series of viruses. And then there's the beta coronavirus, common coronaviruses that were also included in this platform. And the reason they included all of these viruses, it's an opportunity to really screen for the different types of epitopes that are recognized by the antibodies in the plasma from people that have recovered and were exposed to coronavirus, to the SARS CoV-2. So this is the first figure in this paper, and this is his first identification here of the reactivity of antibodies to the different coronavirus peptides. As I mentioned, this is the IgG response only that he's looking at. On the left, you can see the different viruses that are included in the VirScan. And on the bottom of each of the panels, you'll see on the left there's a COVID-19 patient plasma. There's 232 patients included here. And on the right are the pre-COVID era controls, some of which we contributed to for this work. The heat map here is colored by peptide hits from each individual for each of the different epitopes that were that provided a signal in his VirScan assay. So the top row is SARS CoV-2 specific epitopes or peptides, and you can see in the patients clearly that there's a very strong signal for many of the individuals. And again, if you look at the color code on the right, those are peptide hits. So the bluer the color, the more hits you get for each of the epitopes. But you can also see that for SARS-CoV and MERS, you can start to see also signals in patients that have been exposed to SARS-CoV-2. And this is where they describe later in the paper that there is cross-reactivity of the antibodies in a COVID patient to these other related viruses. Now, interestingly, they included the bat coronaviruses, which, as we know, are more related to the SARS-CoV-2 than the other common coronaviruses. And you can also see there are positive hits in that in plasma from those COVID-19 patients. And then on the bottom, you can see these are the common coronaviruses compared directly to the SARS-CoV-2 peptides, and you can see that there are some hits, but in general, they're very low in compared to the related viruses. Now, if we summarize this data from his VirScan technology, you can see quite clearly on the left that those related viruses shown here are showing increased peptide hits in COVID-19 patients as compared to the negative controls, the pre-COVID samples. Whereas on the right, you can see that the common coronaviruses, there is some increase, but it's not nearly as high as what you see in the more highly related viruses. So not super surprising, but also it was important to actually document this. And this is really one of the few ways where you could do a full analysis of the cross-reactive epitopes that are recognized by plasma from patients that are actively or have been convalescing after COVID infection. So this is the next step in the analysis. And what he did here, or what his group did, is they looked at the distribution of antibody reactive peptides among the different SARS-CoV-2 open reading frames. So as in that first figure, I showed you the map basically of the SARS-CoV-2 virus. This is all displayed here on the left-hand side of the figure. And I think it's very easy for us to appreciate that in this figure that most of the peptide hits are in the spike protein and the nucleoprotein. So these are the immunodominant proteins of the virus. But surprisingly, it was also shown that there were hits in the open reading frame 1, which is a very large open reading frame in the SARS CoV-2 virus. And you can also see that in the pre-COVID samples, there's also some positivity against this, this ORF1. And this is likely due to the fact that this is a structural and very essential part of the coronaviruses, and it's also more homologous among the different viruses. So now we get into where we started thinking about using Luminex technology. So as I mentioned, we had a lot of Zoom calls during this time. And I had heard from Dwayne Westerman that Steve was interested in trying to develop a high throughput and kind of a validation translational approach to his VirScan identifications, because when you're screening for using the VirScan technology, it is not necessarily non-labor intensive. It actually is a lot of work to do the studies, and it's really not set up for high throughput diagnostics. So Eric Fujimura in his lab is actually very good at computational biology, in addition to being very good at working with his hands at the bench. So what he did is he took the information from the VirScan platform and he used something called gradient boosting machine modeling to identify peptides that could help diagnose and discriminate between people that are actually have been exposed to coronavirus versus those that are pre-COVID or not exposed to the virus. And the way he had to do this is he had to treat IgG and IgA immunoprecipitate information separately. He put it into this modeling approach, which is, I'm not a computational biologist by any means. We actually are starting to use this, but this is truly a machine learning or a training approach to identify and basically bin positive and negativity through a decision tree approach. He then took this information, put it together into this thing called Ensemble, where he could put the IgG and IgA together, and then came up with top peptides that one could potentially use for a diagnosis of not only active infection, but potentially to use for determining the phenotype of the patient, whether they're going to have a severe infection or a mild infection. And those top hits are shown on the right here in panel C. This again is like I said, it's a kind of a ranking or a testing procedure where you use the data as training and then testing. But through a number of iterations, these are the top hits that he discovered that could be potentially used to make this diagnosis. And then on the bottom left is a visual display of this machine learning output, showing the IgG, IgA, and the Ensemble model. And you can see the percent calls in this, what they call a confusion matrix. You can see that you can tell that the actual diagnosis and the predicted diagnosis are very close. And then finally, importantly, if going back to the VirScan data, they were able to back calculate this and display the actual results for each of the top hits of the peptides and show the actual VirScan data. And you can see quite clearly that these peptides are really, really important immunodominant epitopes and also potentially useful for developing Luminex assays or other types of ELISAs that one could use to diagnose patients. So in discussions with Steve over the phone, he basically posed this question to me, like, what would I do to potentially take the peptide information that he has from the VirScan and develop an immunoassay? He was thinking of actually taking the information and putting it into a yeast and then using that as his platform for the peptide translation. And I said, well, I bet you we could probably do something with Luminex bead technology. So I went to literature and I thought about it. I'd never worked with peptides before. It was all new to me. Most of our work has been done with proteins or nucleic acids. So I found a paper where they described something like this, where they were able to put peptides on the beads. So this was our approach that we tried and came up with to actually couple these peptides that were immunodominant and potentially diagnostic for SARS CoV-2 onto Luminex beads. So the first step was to functionalize the beads using the standard sulfo NHS and EDC chemistry and using an amino PEG azide, and decided to add the PEG in there to make it more soluble and flexible, so that if the peptides were going to be binding specifically, they would have some movement on the bead. We then use click chemistry reaction, which is terrific because it can be done in aqueous, it can be done with pH independent, and it's also very easy to synthesize your peptides with the alkyne modified end. So specifically you're binding that peptide to the azide domain of the PEG4, amino PEG4 that's functionalized on the bead. So in our first attempts, we used about 25 different peptides, and they're listed here. They're from the different domains of the virus. This was all done actually before he used the predictive model. So, science is sometimes a lie. You say, oh, you came up with this great approach, but sometimes you gotta go backwards and forwards to figure things out. So in the end, we developed 25 different assays using this technology, and then focused then on those 12 that were identified as being the most predictive. So here's some things that were very unique to developing assays for SARS CoV-2. So first of all, we were not allowed to work in the lab, work on plasma samples, unless we provided a way to kill the virus. So one of the things that was discovered early is Triton X100 will kill the virus, any live virus. So we had to come up with a assay buffer that used Triton X100, which turned out to be very easy. We were able to use that instead of Tween 20 in all of our buffers. We also had to do a pre-inactivation of the plasma in order to satisfy the biosafety committees at our hospital and at Harvard. The other thing that we did was, because we were thinking of making this a diagnostic test, is that we reduced the incubation time from the higher incubations that we use for immunoassays to 30 minutes. And actually, in the end, we went down to a 20-minute incubation time for all of these assays that I'm going to be showing you. And then, of course, we had to do all the titrations. We had to get concentrations right for the detection antibodies. And this was, of course, to reduce any assay background. Now, I will tell you, initially, we started looking at IgM, IgG, all the IgG isotypes, and IgA, including the two different isotypes. And in the end, we discovered that really trying to get too specific with this approach is probably wasn't worthwhile. So we then focused back on doing just the whole IgG molecule and the whole IgA, even though we had data on the A1 and A2 and the different 4 isotypes of IgG. And then importantly, we found that IgM really provided a very high background. We could still detect binding with the patients, but the background was really a difficult issue. This is something that we identified not only in the peptide assays, but in also whole protein assays that we developed for a number of the SARS CoV-2 proteins. And then my interactions with others that have used the beads that showed that also had the same issues with the IgM, and even using approaches like urea stripping really did not help. So these are some of the tricks that we just wanted to share with you, some of the tricks that we had and things that we learned. Now, importantly, rather than ELISAs, as you know, you can multiplex these assays. So because of that, we were able to get a lot done very quickly We multiplexed the 25 different beads in one reaction, and then in the end, after doing the separate IgG or IgA reaction, we could then pool them back together and then run them on the instrument in one well. And this obviously was saved a lot of time and effort, and also, I think, increased the accuracy of the assays. And as I mentioned, we did all these assays on the xMAP instrument, and we used enough beads in the assay so that we could get reads of around 20 seconds per analyte. And this was just in an idea at the time as a goal to potentially develop this into a diagnostic test for COVID. So this is a summary of the 12 peptides that were identified by machine learning. Summary of the data generated with Luminex. On the left, this time is the pre-COVID patients samples. There were 165 included here. On the right are the 163 COVID-19 positive patient samples. And I think you can really clearly appreciate that we have a very strong response in the COVID-19 patients, but lower responses in the pre-COVID. However, there were some positive reactivities there, which we don't know exactly why, but obviously it's cross-reactive antibodies. They could have had some other coronavirus infection recently that boosted the particular antibodies. As mentioned, most of the peptides are in the S, or the spike, and the nucleoprotein. However, there were a couple you can see on the right, ORF1, where we included that in the assay, and also ORF3A, which were also very positive hits in the VirScan. Importantly, we had controls. They included peptides for Rhinovirus A and Epstein-Barr virus, and you could see very, very high levels of antibodies against these common viruses that are in the general public. Steve referred to these as public epitopes, which I think is a great way to put it. Highly dominant, immunodominant epitopes or peptides from these viruses. And then as a negative control, there's a HIV peptide that was included in the assays as well. Obviously, I was not concerned, but I could see that there were a couple of people that are positive here for HIV, but overall it was a very low signal. And then in this particular figure, we're showing some very important data. Really, is we've used these assays and we wanted to see if people that had COVID-19 that were not hospitalized versus those that were hospitalized. So looking at severity, you can see by using the Luminex platform, you can clearly see that those that were hospitalized had a much stronger antibody response. Now that may seem like a very obvious result, and it is obvious. But what's also interesting to me is that these people that were exposed to COVID, that were not hospitalized and had COVID-19 infection, many of them had low or no antibody responses against these important virus epitopes. So clearly, just getting an infection and recovering from the infection or being exposed is not enough to gain a robust immune response that could potentially protect you from reinfection. And of course, that's why vaccines are developed. And you could see that the assay performed very well for the rhinoviruses and the Epstein-Barr virus. And there were some positivities for HIV, but in general, our negative control worked very well. And then finally, after we generated all this data with the Luminex assays, Eric went back and performed his computational analysis and found that not only did our assays work, but by using these selected peptides, we're able to get very good results with high predictive probability for COVID positivity, very similar to what they found in the VirScan. So just as a summary and a reminder that if you want to learn more about this work, it was published last year in Science. Some of the other notable observations in this paper was that this technology recognized more than 800 epitopes in the SARS CoV-2 proteome that were reactive against antibodies, including 10 epitopes that are known to be neutralizing reactive. Pre-existing antibodies and controls recognize the ORF1 of SARS CoV-2, as I mentioned, whereas only the COVID-19 patients primarily recognize the spike and the nucleoprotein. And that's important, so the vaccines should be focused on the spike and the nucleoprotein, and of course, these effective vaccines are focused on the spike protein. And then individuals with more severe COVID responses did exhibit a much stronger antibody response, but they also had a broader response against the different epitopes. Again, just exposure and maybe not a less severe exposure or less severe reactivity to the virus does not always lead to an effective antibody response. And I think the data really clearly shows that. So in closing, I just want to acknowledge those individuals that really I've mentioned throughout the talk, that where we worked on this together through the MASS Consortium on Pathogen Readiness, Steve Elledge and Dwayne Westerman in particular, and then the two research fellows in Steve's lab that did all the VirScan work, Ellen Schrock and Eric Fujimura, and of course people in my lab who helped me throughout the process, not only with assay development, but also with data analysis. And I will say that there were many contributors to the Massachusetts Consortium. It wasn't just these two guys. Did facilitate many collaborations, and it was really a fun time for scientists. It was a terrible time for the world, but it was a great time for everyone to actually contribute and get together and do our best to try to help solve the problems. And of course, I have to thank my funding sources that supported me during this period of time.